আমার আসবে ওরা পোস্টারে সবজন আছে
মেসেজটা তো দেওয়া রিজাল্ট ক্যামেরা রিজাল্ট ঘুমাবে যারা আছে নিজেই এডমিট হয়ে যাবে
তিনজন আছে মাস্টার মনে আছে যে ইউকে থেকে উনি আমাকে পাঠিয়েছেন দিল্লি এখন দিদি চেঞ্জ করে দিতে হ্যাঁ ও হচ্ছে মগজের ওনাদের টাইমটা ওনাদের টাইম তো হচ্ছে না ওই জন্য এখানে কোটাই ওরা হচ্ছে ওই জন্য আমাদের পিপিটি আমি পাঠিয়েছিলাম উনি মোবাইল থেকে হচ্ছে 4:30 পাঁচটা অবধি যাবে
আজকে 5টা শেষ হয়ে যাবে
5টা শেষ হয়ে যাবে আচ্ছা হয়ে গেছে স্টার্ট করে দেবো
স্টার্ট করবো ক্যামেরাটা ঘুরে করে নি
না না করিনি ঘুরে দিন
খালি
এই একটু আগে উপরে আছে নাকি
শুধু টিচার যেন আছে কেউ আসছে না ওইখানে পোস্টার চলছে
पोस्टर सेक्शन में जितना भी है ना पार्टिसिपेंट्स उनको इधर में भेज दीजिए ना रूम नंबर 1 जो हो गया हाँ पोस्टर सेक्शन का पार्टिसिपेंट का यदि उन लोग का हो गया होगा तो इधर भेज दीजिए ना रूम नंबर 1
Good afternoon, everybody. A very warm welcome to our eminent speaker. A very good afternoon to everybody. A very warm welcome to our eminent speaker, Dr. Jackson D'Souza, Associate Professor, Sao Paulo State University, Brazil, delegates and participants to the second day of our international seminar, Technical Session 2. I am greatly honored to introduce today's eminent speaker, Dr. Jackson D'Souza, who is going to speak on his title, The Next Bio-Resource Frontier, soil, ecogenomics, and biotechnology for sustainable agriculture and environmental remediation. Dr. Jackson D'Souza holds a BSc in Biological Science and an MSc in Biosciences and Biotechnology with a focus on genetic engineering from the State University of Northern Rio de Janeiro. He earned his PhD in agricultural microbiology in 2006 and completed a postdoctoral fellowship in 2009 at the School of Agriculture and Veterinary Science, Sao Paulo State University, where he serves now as associate professor. Dr. D'Souza is involved in ecologically oriented projects related to biological resource management, bioenergy production, second generation ethanol, bioremediation, and environmental impact and development in agricultural systems. He is a member of the Brazilian Advisory Board, Southeast region of the Brazilian Microbiome Project and of the BIOS Brazil Research Network. He has participated in the National Institute of Science and Technology for Biological Nitrogen Fixation in 2014 and is currently a member of the INCT for Soil Biodiversity, Biotechnology and Sustainable Use of Biodiversity theme since June 2019. He has chaired the Permanent Committee for Genetic Heritage Management at UNSP. Dealing with issues related to genetic heritage, biodiversity, and associated traditional knowledge. We humbly invite him to our seminar today, and sir, the dais is all yours. You can start with your presentation. Thank you, sir.
Thank you for this kindly presentation. I will share my presentation now. All right. Could we get started now?
Yes, sir, please.
Okay. Good afternoon, good morning here in Brazil. I would like to thank you, the organizing committee of this international conference, particularly to Dr. Swat Shakabort, my colleague, for this invitation to speak with you today. It's truly an honor to show you data from my work in Brazil, a country rich in agriculture, heritage, and always searching for innovative, sustainable farming. I'm a research assistant professor at São Paulo State University in Brazil, where I work on applied genetics and molecular microbial ecology. I'm sorry about this terrible voice, but I got COVID this 2 days ago. Today I will share with you what I believe is one of the most promising frontiers in agriculture science: soil epigenomics and microbial biotechnology. We'll be exploring how the invisible world beneath our feet, the soil microbiome, holds solution to some of the most pressing challenges: feeding a growing population, reducing environmental pollution, and combating climate change. Specifically, I will share insights from our research in Brazilian sugarcane system, which serves as a model for sustainable agriculture globally. Let's start with the reality we're facing. By 2050, we needed to produce 70% more food than we do today. But here's the catch. We're running out of farmland, and climate change is making farming increasingly unpredictable. Now look at these numbers. 1/3 of our global soils are already degraded. The very foundation of our food system is eroding, losing nutrients, and becoming less productive. This isn't just an environmental issue, it's an economic and food security crisis. Here, another critical problem. When farmers apply nitrogen fertilizers, which are essential for crop growth, 50 to 70% of it never make it into the plant. It either leaches into groundwater, causing pollution, or volatilizes into the atmosphere. This is an enormous waste of money and resources for producers. And perhaps most concerning from a climate perspective, nitrous oxide, or N2O, is 300 more potent as a greenhouse gas than carbon dioxide. Agriculture is a major source of this emission, particularly from nitrogen fertilizer use. There aren't separated problems. They're all connected. They're symptoms of soil ecosystem dysfunction, and that's where microbial solution comes in. Now let me introduce you to the healing word that can solve these problems. In just 1 gram of healthy soils, there are more than 1 billion bacteria. That's more microorganisms than there are people on Earth, all working together in complex communities. But here's the contradictory part. 99% of these microorganisms we cannot grow in our laboratories using traditional methods. We call this the Great Plate Count Anomaly. For over a century, microbiologists could only study the 1% that grew on petri dishes. These microbes aren't just passive residents, they're the living engine of nutrient cycling. They transform elements like nitrogen, phosphorus, and potassium from forms that plants cannot use into forms that plants can absorb. Without them, modern agriculture would be impossible. They also provide what we call ecosystem services. It means building soil structure, helping plants to survive drought and salinity stress, and suppressing pathogens diseases without chemicals. Here, the omics revolution changed everything. Using modern DNA sequencing technologies and metagenomics, we can now discover the genetic blueprint for entering microbial communities directly from soil samples. We're no longer limited to the 1% we can filter. This represents a fundamental paradigm shift. We're moving from simply asking who is there, identifying species, to asking what are they doing, understanding their metabolic function and how they contribute to soil health and crop productivity. Okay, so why do we study sugarcane system? Brazil produces 4% of the world's sugarcane, making it a global agricultural powerhouse. But sugarcane is much more than sugar. It's a renewable champion, producing bioethanol as a sustainable alternative to fossil fuels. However, this intensive production comes with environmental challenges. For every liter of ethanol produced, we generate 10 to 15 liters of vinasse, a dark, nutrient-rich liquid residue, beside massive amounts of straw, bagasse, and filtered cake. These residues aren't waste, they're opportunities. When returned to the soil, they dramatically reshape microbial communities. They create what we call nitrogen hotspots, moments and locations where microbial activity explodes, transforming nutrients at high rates, but also potentially releasing greenhouse gases. This makes sugarcane system a perfect natural laboratory. They're economically important, environmentally challenging, and microbially dynamic. Understanding how microbes respond to these intense management practices gives us insights into applicable to agriculture worldwide. To give you geographic context, in the 2023-2024 harvest season, Brazil produced over 730 million tons of sugarcane. More than half of this comes from São Paulo state, the region where our research is focused. This concentration means São Paulo has the highest density of mills and the most advanced agricultural technology. The three strategic outputs: sugar for global export, bioethanol for renewable energy, and bioelectricity generated from burning bagasse, make this system critical for both Brazil's economy and global sustainability efforts. Let me show you the fundamentals of the denitrification pathway, especially because this is where greenhouse gas emissions primarily occur and where we see the greatest potential for microbial intervention. Denitrification is a four-step process, each controlled by specific enzymes. We start with nitrate reduction to nitrite, catalyzed by the enzyme encoded by nitrate reductase gene. This happens when soil becomes waterlogged and facultative anaerobes switch their metabolism to use nitrate instead of oxygen. Then nitrate is reduced to nitric oxide by enzymes encoded by nitrate reductase genes. Then nitric oxide reductase reduces it to nitrous oxide, and this is our major problem molecule. N2O is 300 times more potent than CO2 as a greenhouse gas. But here's the critical point: there's one more step. The nitrous oxide reductase enzyme converts N2O into N2, an inert atmospheric nitrogen. This is nature's environmental solution, the major N2O sink. This is our biotechnology target. If we can introduce microbes with robust NOS-Z genes or manage soils to favor NOS-D carrying bacteria, we can turn agricultural soils from greenhouse gas sources into sinks, completing the denitrification pathway. Environmental controls matter too. Acidic soils often inhibit NOS-Z enzyme activity, as does low carbon availability and certain C2N ratios. Understanding these controls allows precision management. Concerning agricultural fields, into the nitrogen biogeochemical cycle, we have some inputs of nitrogen as synthetic fertilizers like urea and ammonium nitrate, plus organic residues like vinasse and filter cake. These provide nitrogen in forms microbes can work with. The problem in many agricultural soils is pathway imbalance. We have plenty of the early pathway genes, lots of microbes producing N2O, but insufficient NOS-Z activity to complete the reduction. It's like having a production line where the last quality control station is understaffed. The transformation pathways depend on soil conditions. particularly oxygen and moisture. When soil is well aerated, we get nitrification process, where ammonia is oxidized to nitrate. When soil is waterlogged, we create anaerobic conditions that trigger denitrification. The carbon to nitrogen rate also matters to drive the cycle in favor of immobilization. Microbes incorporating nitrogen into their biomass or mineralization, releasing nitrogen to plant use. During nitrification and denitrification, if the conditions aren't perfect, it leads to environmental leaks. We get emissions of N2O and nitric oxide, greenhouse gases, instead of the desired end product, inert N2 gas present. We can also lose nitrate through leaching. Nitrate is negatively charged, so it doesn't bind to soil particles. When it rains heavy or we over-irrigate, nitrate washes through the soil profile into groundwater, causing pollution and representing economic loss to farmers. Understanding these pathways and their microbial drivers is essential for developing management strategies that maximize and minimize environmental impact. Regions with intensive agriculture, particularly in São Paulo state and southern Brazil, show direct correlation between nitrogen fertilizer application and atmospheric emission peaks. The primary driver is agriculture. Intensive use of synthetic fertilizers combined with inefficient nutrient management creates massive nitrogen surpluses. The application of both synthetic fertilizers and organic residues creates the already mentioned hot moments of nitrogen availability, times when microbial activity spikes and emissions peak. The urgency is clear. We need to biogeochemical flows through precise microbial management and nitrogen loss reduction. This isn't optional anymore. It's critical for planetary stability. And this is where functional microbial inoculants come in. Targeted biological solution that acts perfectly during these activity peaks to redirect nitrogen transformation toward productive environmental healthy pathways. So how do we develop these solutions? Through what we call the ecogenomics revolution. Traditional soil microbes relied on culture, growing microbes on petri dishes, for example. This gave us important information. We could isolate specific organisms, characterize their metabolism through laboratory tests, and study their phenotypes. But remember, this only captured about 1% of soil diversity. Metagenomics changed everything. We extract all DNA directly from soil samples and sequence it using high-throughput technologies. This gives us a comprehensive profile of the entire microbial community, including the uncontrollable 99%, and allows us to mine for functional genes without ever growing the organism. But sequencing alone isn't enough. We need functional ecology, linking genes to actual ecosystem process and management outcomes. This means measuring process rates and fluxes in the field, using predictive modeling to understand how communities respond to management, and finally, enabling precision agriculture. Our approach integrates all three of this process. We use metagenomics to discover functional diversity, culturing to isolate promising strands, and functional ecology to validate performance under real agriculture conditions which enable targeted bioresource development. Let me show you our specific methodology, which combines culture-independent approach and culture-independent approach. On the left, culture-independent methods, we use enrichment techniques like vinogradicus columns, vertical gradient systems supplemented with different nitrogen forms, to selectively grow functional groups like nitrifiers and denitrifiers. We screen these isolates for plant beneficial traits, phosphate solubilization, production of growth hormones like auxin, and ability to grow in harsh conditions like high vinasse concentrations. Finally, we validate candidates through greenhouse trials and field experiments on sugarcane, maize, and garlic. On the right, culture-independent epigenomics. We perform shotgun metagenomic sequence on total DNA from different environments, sugarcane soils, filter cake compost, and others for comparison. We reconstruct, sorry. We reconstruct metabolic pathways computationally, focusing particularly on nitrogen metabolism. And we mine for specific biomarker genes. A for ammonia oxidation, NIR-K and NIR-S for nitrate reduction, NOS-Z for N2O reduction, to link taxonomy with nitrogen cycle functions and greenhouse gas emissions. Integration for management decisions is the utmost result. Metagenomics tells us what functional potential exists in the community. Culturing gives us strengths we can work with. Functional ecology validates which approach actually work under field conditions. Now let's look at our first case study, sugarcane soil, which we abbreviate as SSCP. This is a typical agriculture field with mechanized harvest and straw retention. We sample both harvested soil near roots and intergrow soil from the top to end centimeters. Each harvest leaves 10 to 20 tons per hectare of straw on the surface, and farmers typically apply 6 to 120 kilos of nitrogen per hectare. The microbial community here is what we call autochthonous, well adapted to local conditions. It's dominated by moderately coprotrophic groups, organisms that respond to nutrient pulses but aren't extreme fast growers. The functional diversity is lower than in native forest soils due to intensive management. The management regime creates frequent nitrogen hotspots. High synthetic nitrogen inputs combined with organic amendments like vinasse and filter cake create conditions for rapid microbial transformations, which can be productive for plant growth or problematic for emissions, depending on the balance. Nitrogen dynamics of this soil condition was explored by metagenomic analysis and reveals the functional profile. The microbial communities show strong ammonification, high capacity to mineralize organic nitrogen into ammonia. This is good for releasing nitrogen from organic matter. However, the nitrification potential is relatively lower, about 2.3 times less than filter cake composting, which we will discuss shortly. But here's the critical finding: pathway imbalance. Look at the functional genes profile. We see abundant genes for the early denitrifying steps, producing Na2O, but limited NOS-Z genes for the final reduction step to inert Na2. In this case, the intermediate is nitrous oxide, a potent greenhouse gas. Management practice influences this. The straw cover significantly increases microbial biomass. More microbes mean more potential activity, but straw also retains moisture and can create anaerobic microcytes under the residue layer. This favors denitrification, genes like NIR-K and NOR-B. The implication is clear. We need NOS-Z enriched in inoculates. If we can introduce bacteria carrying robust NOS-Z genes that persist in this environment, we can complete the denitrification pathway and reduce greenhouse gas emissions while maintaining productivity. Our second case study is filter cake, abbreviated as CIT, a very different environment. Filter cake is an industrial byproduct from sugarcane processing. During juice extraction, the juice is filtered, leaving behind press mud, a wet, nutrient-rich material. This has about 7% moisture, nearly 2% phosphorus on a dry basis, high organic matter, and is rich in calcium and nitrogen. In practice, farmers apply 15 to 30 tons per hectare. This significantly improves soil properties, increases cation exchange capacity, the soil's nutrient holding ability, improves water retention, and provides essential micronutrients. We studied in an immature composting state, not truly decomposed yet, still with active thermophilic communities. The physical-chemical environment is distinctly alkaline around pH 8, which is important because pH dramatically influences microbial community composition and activity. The substrate is extremely rich in organic matter, serving as fuel for intense microbial activity. This creates a dynamic community in transition, high proliferation rates, rapid ecological succession, and selection for groups adapted to alkalinity and nutrient abundance. Filter cake compost is the most metabolic active system we study for nitrogen cycling. It has approximately 2.3 times more higher denitrification potential than sugarcane soil, with very high nitrogen turnover rates. But here we found an interesting result. The emission signature is different. In sugarcane soil, the primary concern is N2O. In filter cake, it is nitric oxide, NO. The combination of alkaline pH and the specific microbial community structure favors NO accumulation over N2O. This is still an environmental pollutant and represents nitrogen loss, but it's a different management challenge. This reinforces why ecogenomics is so powerful. We can diagnose the specific functional imbalances in each system and design targeted interventions. Comparing communities, we can see these two systems to understand the ecological strategies. In sugarcane soils, SCP, we see oligotrophic dominance. There are bacteria adapted to low nutrient availability and acidic pH, like acidobacteria and verrucomicrobial. The community has high diversity but a lower overall biomass compared to filter cake. In filter cake, CIT, we see copiotrophic bloom, rapid responders to high carbon and nitrogen inputs. Proteobacteria, firmicutes, and bacteroidetes dominate. These are fast-growing decomposers with high metabolic rates. For nitrogen cycle, in SCP, the key process is nitrification, oxidizing ammonia to nitrate, driven by fertilizer inputs. We see high abundance of amyloid genes and stress response genes, reflecting the challenging fluctuation conditions. By the other hand, the nitrification and the NRA, dissimilatory nitrate reduction to ammonia, dominates CIT. We find enrichment of NARD, NIRK, NS, and importantly, NOS-Z genes, the N2O reduction capability we wanted to manage. The keystone taxa in soil, we find specialists like nitrosphere and red rhizobium, critical for basal nutrient cycle. In filter cake, we find metabolic generalists like Pseudomonas, Bacillus, and Streptomyces, producers of enzymes that break down complex organic residues. Here is the management insight. When you apply filter cake to sugarcane soil, you're essentially performing a microbial inoculation event. You're shifting the soil community toward a more copiotrophic state, accelerating nitrogen turnover. Understanding this allows strategic timing and application rates. So we have identified the problems and strategies. Now let's talk about solutions. We screened for PGPR, plant growth promoting rhizobacteria, with specific target traits. Phosphate solubilization. We look for strands producing halos up to 5.5 millimeters on indicator media, showing they can mobilize anorganic phosphorus. Often the most limiting nutrient despite high total soil phosphorus. Production of indolacetic acid, synthesis of disoxin, a plant hormone that stimulates root growth and architecture. Nitrogen cycle modulation, ability to regulate nitrification and denitrification steps, potentially reducing losses. From our isolation efforts across sugarcane and garlic rhizospheres, we identified promising genera, Penibacillus, Pseudomonas, Bacillus, and Brevibacillus. When we analyze co-occurrence networks, we compare garlic soil that is near the sugarcane crop area. Comparison of bacteria associated with each other, we found a dramatically higher complexity in garlic soils compared to monoculture sugarcane. This suggests garlic systems, which often involve crops rotation and organic inputs, maintain more resilient, functionally redundant microbiomes. This gives us clues about which consortium designs might work best. Now let me introduce you to our bacterial strands isolated from those different crop soils that show the significantly promise both in laboratory assays and field trials. First of all, our top performer, Penibacillus alveoli, a spore-forming rhizobacterium from the Firmicutes phylum. We recover the multiple strands showing strand-level functional variation. We highlight particularly strain AN31, It's our top performer for phosphate solubilization with 5.5 milliliters halos, meaning it very efficiently mobilizes insoluble phosphates through organic acid production. It also produces IAA stimulating root growth. In trials, AN31 improved sugarcane shoot biomass and both garlic root mass and length. For garlic strength, TSB02 significantly increased fresh root mass, and strain TY90 improved root length. But here is the critical challenge. Vinasse tolerance. Strain IN31 tolerates up to 25% raw vinasse concentration under acidity conditions and up to 50% at neutral pH. Vinasse is harsh, pH around 4, high chemical oxygen demand, phenolic compounds. Most bacteria can't handle it, but these penibacillus strains not only survive, they thrive, meaning we could potentially produce it using industrial waste as a low-cost growth medium. Other isolates spotlight goes to Brevibacillus, Bacillus, and Pseudomonas strains due to their diverse functions. Brevibacillus nitrificans strain EV08 is our field leader. In field trials, it produces the highest shoot biomass. We will see the data shortly. Like Penibacillus, it thrives in raw vinasse environments, making it suitable for circular economy formulations. It shows both nitrification potential and excellent adaptability to sugarcane industry byproducts. Bacillus species, particularly BioEd4 and BioEd5, show remarkable diversity. BioEd4 demonstrates the nitrifying activity, consuming nitrite, which makes it valuable for modulating the dinitrification pathway. BioEd5 grows robustly in nitrifying media and shows exceptional viability in culture, producing high colony form units or CFU counts, making it ideal for commercial inoculant formulation. Pseudomonas species like TW21, isolated from garlic soils, is a gram-negative PGPR candidate. The Pseudomonas genus is legendary in biocontrol and growth promotion. TW21 showed strong phosphate solubilization and IAA production, though we haven't yet tested it as extensively in sugarcane. These strains consist of a promising candidate for consortium design. The selection criteria combine viability, can we produce it efficiently, with biomass efficacy, does it actually promote plant growth under field conditions, and metabolic versatility. Can it function across variable environmental conditions and contribute to nitrogen cycling? Now for the proof, field trial results with sugarcane 30 days after inoculation. Look at these bars. Brevibacillus nitrificans, EV08, was the clear winner. Compared to uninoculated controls, it increased shoot fresh mass by 38% and sugar dry mass by 42%. The error bars represent 95% confidence intervals across 5 field plots are small, showing low variability and high consistency. These aren't just statistically significant, they're agronomically meaningful. Increased dry matter accumulation indicates enhanced carbon fixation and nitrogen assimilation efficiency. Remember, sugarcane is harvested for biomass. More biomass means more sugar, more ethanol, more bioelectricity. This validates EVO8 as a robust candidate for commercial biofertilizer formulation. The consistency is crucial. Farmers need reliability, not just peak performance under optimal conditions. In summary, let me show the highlights for our elite candidates. Brevibacillus nitrificans strain EV08, dual function, root development plus nitrification potential, besides good potential for vinasse tolerance. Penibacillus alveoli, ADN31, shoot biomass, promotion with highlighted vinasse tolerance. And Bacillus wedgmenti, strain by WED 5. Besides other functions, show exceptional CFO production, which is ideal for commercial formulation. Together, these isolates represent a functional toolkit. Some are growth promoters via hormone and phosphate. Some modulate nitrogen pathways. Some tolerate harsh waste streams. Depending on the application, whether it's through its nutrient efficient, emission mitigation, or waste valorization, we can select the appropriate strain or consortium. The strategic insight will be, we're not using single strain. We're designing synthetic communities based on ecological principles. I want to briefly share insights from comparing garden and sugarcane systems. Co-occurrence network analysis looking at which microbes correlate in abundance revealed significantly higher complexity in garden cultivation soils. High connectivity suggests a dense interaction web. In ecological terms, this potentially enhances nutrient cycle efficiency and pathogen suppression through multiple pathways in functional redundancy. This indicates a more resilient microbiome, one capable of sustaining plant health under stress, because if one functional group declines, other can compensate. The top 13 enriched genera identified in these high-performance soils give us ecogenomic targets for future isolation efforts, and we're essentially learning from naturally high functional systems to engineer improved looks. We can see other functions not mentioned until now, such as degradation of aromatics, nitrogen fixation potential, adaptation to arid and low nutrient soils, polysaccharide degradation, by example. Let me tie this all together with our next generation bioresources framework, a closed-loop approach from discovery to field implementation. The first step, ecogenomics engine. We mine metagenomics for nitrogen and phosphorus cycling genes to understand functional potential. We use this information for target isolation of Penibacillus and Bacillus strain. We screen for traits like vinasse tolerance and that match our production constraints. The second step, think that communities. We don't just throw handle strengths together. We use network topology, the co-occurrence patterns we discussed, to design consortia that mimic naturally high functional communities. We ensure functional redundancy, so if environmental conditions change, the consortium remains effective. Bioprocess scale-up, the third step. This is where circular economy comes in. We use waste materials like raw vinasse as culture media, dramatically reducing production costs. We explore integrating production with filter-taking composting. This transforms waste management problems into solutions. Finally, the fourth step, digital agronomy. We develop decision support tools, data-driven recommendations for application time based on weather, soil conditions, and crop stage. We integrate with precision fertilization system farmers already use. We track inoculant persistence using molecular markers to understand long-term effectiveness. Sorry. The key outcome is bridging the gap between metagenomic discovery and on-farm application through circular bioeconomy principles, ensuring both economic sustainability and environmental efficiency. This isn't just laboratory science. It's a complete framework for translating genomics insights into agricultural practice. So let me conclude. We're at a pivotal moment in agriculture science. The invisible world of soil microbiomes holds solutions to climate change, food security, and environmental pollution, but only if we can harness it intelligently. The research I've shared today from the Brazilian sugarcane system is just one example, but the principles apply globally. Every agricultural system has its own microbial potential waiting to be unlocked. Thank you for your very much attention, and I'd like to thank my research group in Brazil and financial support agencies. Thank you.
Thank you, sir, for your enriching talk. Should I open the house for interaction, sir?
Hello. Yeah. It is Swati. After a long time, Professor Jackson's.
Hi.
Yeah. So it was excellent presentations, and after that Brazil. So I have got an opportunity to interact with you also. I have one questions to you regarding the consortia designing. Why you are like what factor like you have considered that to take the garlic as a like as a plant and as a and another source is the sugarcane. Why it is a garlic? Like why you have chosen the garlic as a, another plant source?
Sorry, but I can't hear very well your question. I don't understand because the sound is not so good.
Yeah, actually I have a questions like as you have chosen the garlic as a consortia
Yes.
that with the comparisons with the sugarcane. Why it is a garlic? Like in which point of like the parameter you have considered for that? Like that to have the garlic and other the comparison factors with this. And I have another questions also, like in the sense of making the consortia, there are the different types of PGPR activity you have taken. So, like, what are the quantifications? Like, how you have quantified, like, IAA productions and all that, like, phosphate solubilizations, like, how the quantifications part you have considered in the making of the consortia, why you have made the consortia?
Okay, I didn't explain, but in our area of study, the field is primarily used for sugarcane crop. But particularly in this area, we have like a pocket of garlic field surrounded by sugarcane, all right? And the farmer that produces garlic is well succeeded in this production because he got good condition for garlic production. In our state, São Paulo, garlic is not a good plant to grow because it's very warm. They produce garlic in Minas Gerais. So this open our mind to focus on this garlic food, thinking in this promising opportunity to get bacteria and genes that was interesting for both cultures. Okay? It was the question? I don't know if I answered.
Yes, yes, thank you. Hope
Thank
everything
you.
is good on your side.
Okay, Dr. Swati.
Yeah.
Any more questions from anybody? No. Thank you, sir, for your wonderful presentation. On behalf of our college, extend our heartfelt gratitude to you for buying time for us from your busy schedule, and we look forward to a long-term association with you, sir. Thank you.
दूसरा बिंदु बोलिए केंद्र में पीपीटी है
समझ गए तो क्या करोगे
उसको हाँ मिनिमम कीजिए
इधर नहीं इधर
और हम लोग को चेंज करना है ये है
ठीक है तो फिर हम देखेंगे कौन वाला
ये चीज़ चाहिए तो जरा सा कॉल कर दीजिए
এটাই তো আমি বলছি এটাই তো আমি বলছি আমি এখানে কি করে অপারেট করব
এখন থেকে অপারেট করতে বলেছি
এখান থেকে শেয়ার করে দিতে হবে
শেয়ার করে দিই
এখান থেকে প্রেজেন্ট করতে হবে ও জাস্ট বলবে এখানে এক্সটেন্ড করে
এখন থেকে নেক্সট এক্সেস
এই আইডি থেকে শেয়ার করতে হবে স্ক্রিনটা
ওটা ভিডিওর মধ্যে আসবে না
হ্যাঁ
ভিডিওর মধ্যে আসবে না
কেন ভিডিও
আসবে আসবে ভিডিওর মধ্যে আসবে গুগল মিটটা
আরে এই স্ক্রিনটা শেয়ার করা যাবে না এই স্ক্রিনটা গুগল মিটে শেয়ার করা যাবে কি যাবে না এই স্ক্রিনটা শেয়ার করা যাবে তো
করে দেবো
কেন
আর share
এই
করে
প্রেজেন্ট
দিই।
করবে ও শুধু বলবে ও নেক্সট করে দেবে এর চেঞ্জ করবে এই চেঞ্জ করবে হ্যাঁ ও ওই সেটা থাকবে কোণে থাকবে
আমি আসবো না।
আরে না দেখা গেল কোনো অসুবিধা নেই তো না দেখা গেলে কোনো অসুবিধা নেই তো স্ক্রিন শেয়ার করে দাও
শেয়ারই হচ্ছে না
হোল স্ক্রিন ইন্টার স্ক্রিন করো কি অবস্থা হ্যাঁ
তারপর না না শেয়ার হচ্ছে না তাহলে ওটা খুলতে হবে
ও হ্যাঁ
ওটা খুলে রাখতে হবে
হ্যাঁ ওটা খুলে রাখো
কথা আছে এটা খুলে
না না খোলার সঙ্গে তো ইন্টারেস্টিংয়ের কোনো সম্পর্ক নেই এফআই করো ওকে করো শুধু এফআই করো অল ট্যাবে যাও
এটা বন্ধ হয়ে যাবে
তুমি এবারে ইন্টারেস্ট শেয়ার করো ইন্টারেস্ট ইন্টারেস্ট করো হ্যাঁ ওইটা ক্লিক করো হ্যাঁ এবার শেয়ার করো
স্ক্রিনটা শেয়ার করতে
না 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 হয়ে যাবে চলে আসে
এখানে চলে যাবে
ওই এখানে এবার এবার এই তো ঠিক আছে এই তো ঠিক আছে ঠিক আছে
আপনি বলবেন কি আমরা এখান থেকে শেয়ার করতে বলেছেন আপনি, সো উই আর শেয়ারিং ফ্রম হিয়ার, উইল চেঞ্জ দ্য স্লাইড ফ্রম হিয়ার।
চেঞ্জ করতে হবে এখানে
मैंने दिखाया
ক্যামেরা
মাইক অফ আছে অফ
Hello. Hello. Good afternoon, everyone. It is a great honor and privilege to welcome our distinguished keynote speaker, Professor Michael James C. Gravey, who joins us today in this international conference through the online platform to deliver his keynote address on the compelling and urgent theme, how sustainable are coral reefs when 45% of coral species face extinction. His presence brings exceptional academic depth and global perspective to our deliberation on one of the most critical environmental challenges of our time. Professor Krabbe was a fellow and senior research associate at Upson College, Oxford, part of the prestigious University of Oxford, and a full professor at the University of Bedfordshire. Over a career spanning more than 4 decades, he has distinguished himself as a leading scholar in biochemistry, climate science, computational biology, and environmental sustainability. He earned his BSc in biochemistry from the University of Hull, followed by a PhD and later a DSc degree from the University of Manchester, UK. Professor Kravi's scholarly achievements are remarkable. His research has produced nearly 300 referred journals, publications, and books, along with numerous book chapters and 14 items of commercial molecular modeling software, which was published by Oxford University Press. His expertise in climate change and ecology has earned international recognition. In 2006, he received the 6th Aviva Earthwatch International Award for Climate Change Research, acknowledging his outstanding contribution to understanding the environmental consequences of global warming. He has also been closely associated with conservation and policy bodies, serving as a consultant on a rate listing for the International Union for Conservation of Nature, that is IUCN, where global assessments of threatened species, including corals, are undertaken. Professor Krame's research on the fragility of marine ecosystem, particularly across Pacific island countries and territories, highlights the economic and ecological importance of coral reefs. His work shown the coral reefs support fisheries, tourism, and coastal protection, yet are increasingly threatened by coral bleaching. What makes Professor Grave uniquely suited to today's theme in his interdisciplinary vision. He integrates molecular science, climate modeling, ecological research, economics, and governance into a comprehensive understanding of sustainability. At a time when nearly half of the coral species face extinction, his insights will guide us towards informed action, global cooperation, and responsibilities. We are deeply honored to have Professor Michael James C. Crave with us today and eagerly look forward to his enlightening keynote speech. Professor Crave, we'll show these slides and from here, and we'll check the slides from here.
Lovely. Can you hear me?
Yes.
That's wonderful. Yes, I can see the slide. Thank you very much. That's the first slide. And then if I just ask you to go to the next slide, you can go to the next slide. Thank you very much indeed. Is that okay?
Okay. Okay.
Lovely.
Please go ahead. Next slide.
Right. So it's a great pleasure and honor for me to be able to speak with you this afternoon. You have this afternoon. I'm still in the morning in Oxford. Could we go back one slide, please? Thank you. I'm going to be talking about sustainability, and this relates absolutely to the United Nations Sustainable Development Goals. The 17 goals that are so important that could we go back, please? So important that they should be completed by 2030. I think that's going to be very difficult. Ostensibly, what I'm going to be talking about is number 14, which you can see there in blue at the bottom, life on the water, but actually it reflects on all of them. All these sustainable development goals are united together, and we need to consider all of them, not just one at a time. It's my great pleasure to be a fellow at Wolfson College, a large postgraduate college at the University of Oxford, but I have lots of international collaborations and honorary professors visiting a number of universities throughout the world, particularly Asia. If we could have the next slide, please. Thank you very much. You all know that global warming is happening. On the top left-hand slide there, the top left-hand graph, you can see the temperature increasing. We also have recent sea level rises, that's very important in the ocean. We have at the bottom right ocean acidification, that's a basic chemical process. Carbon dioxide dissolves into the ocean, making it more acidic, and that has very serious consequences for anything that uses calcium carbonate. Skeletons, like corals, like many invertebrates, they become more brittle. Also, the warming ocean means that there are a higher percentage of very strong Category 4 and 5 hurricanes worldwide. So that's extremely important. The hurricanes get their energy from warm water, and the warmer the water, the greater the energy, the more destructive the hurricane. Next slide, please. When you're talking about anything economically, particularly in terms of policy, you've got to think economically and money. So coral reefs roughly have a value of about 10 trillion US dollars globally, and are relied on, and that means for life, by at least 500 million people. They do an awful lot of good. They provide us with medicines, more than half of all new cancer drugs, focus on marine organisms, a lot of biodiversity, they're even more biodiverse than rainforests. They protect the coasts, large coral, massive corals, protect wave energy rushing up into the coasts. They're very important for tourism, economics, and of course, they're extremely important for food security and food production. Next slide, please. One of the things that I'd like you to take home from this talk is ecosystem connectivity. Coral reefs don't just exist by themselves. They're intimately linked to seagrass beds, mangrove forests, and then to the coastal forests. Let me give you an example. Young fish that are spawned on coral reefs are at very great risk of being eaten. So they swim into the seagrass beds, hide in the seagrass beds, often change colour so they can't be observed in the seagrass beds. When they're a bit bigger, they swim on into the mangrove forests, and that helps them be protected by living in the waters and protected against the large predators in the mangrove roots that go above water, of course. They then, when they're sufficiently old and can fend for themselves, they swim back through the seagrass beds, go back to the coral reefs, and spawn on reefs. So that's a wonderful way of thinking about ecosystems. Next slide. Next slide, please. Right, one of the biggest problems that we have is called coral bleaching. We know that temperatures are getting much warmer in the ocean, and warmer temperatures can result in the complete whitening of whole reef ecosystems. Corals are coloured because they contain little plant cells. Corals are animals, polyps themselves, but they have a symbiotic relationship with the zooxanthellae, we call them, the little algal cell plant cells living in their tissues. Those plant cells contain all photosynthetic machinery to allow the corals to utilize sunlight, and so the sunlight then enables the corals to produce sugars and ultimately carbonate skeletons. And the carbonate skeletons are laid down. The colours come from the photosynthetic pigments. The coloured pigments, lots of double bonds in the molecules that provide the colours. But unfortunately, the little algal cells are expelled from the corals when the temperature rises. So they lose their colours, they lose the algal cells, therefore they lose the pigments, and the corals come completely white. Now, corals can survive a bleaching event, but they're under much more stress and are subject to great increase in mortality. I'm going to be talking quite a bit about that later. Could I have the next slide, please? Just to show you some examples of corals, most of these photographs are mine. This is from the Caribbean, reefs around the island of Jamaica. You can see on the top right there a very large and massive coral. Those are the corals that protect the land from wave energies. And then at the bottom, left and right, you can see from branching corals, these are faster growing corals, that again provide important ecosystem for fish. And you can see on the bottom left there, quite a lot of fish around that very vibrant Acropolis cervicornis colony. Next slide, please. In the Indo-Pacific, you've got similar types. You've got branching corals, you've got brain-type corals, massive corals. You've also got plate corals. Those are the three main types. Also in the middle there, you've got a wonderful pillar coral. They're all extremely important. The ecosystem of a coral reef is absolutely vital for all the life, the invertebrate life, the fish life, the microbial life that goes on under the tropical oceans, mostly between the tropics of Capricorn and Cancer. Next slide, please. The big scientific question is how do organisms adapt and evolve in extreme environments? And I did quite a lot of work on that with my colleagues abroad. This is one paper that we published in the Proceedings of the National Academy of Sciences with colleagues at Fudan University in Shanghai, showing that within a C3 plant, you get highly cooperative regulation under changing environments, the sort of environments that you get with climate change, increasing water stress, and also high CO2 conditions. And it's that cooperation, both within organisms and between organisms, that's so vital for the future. Next slide, please. Now, we need to ascertain how much these species are at risk of extinction. That's done with the IUCN, the International Union for the Conservation of Nature, who have a very rigorous process called red listing. And red listing is true for all species on the planet, whether you're an elephant, you're a mosquito, you're a coral, you're a fish, whatever. And that's why it needs to be so rigorous, so you can get some comparison of the extinction risk of all species on the planet. The only species, the only genera that are not looked at in this is microbial. So we don't look at microbes, but we do look at everything else. You can see here birds, mammals, corals, and amphibians, which were looked at from 1980 to about 2012. And you can see here that they're all decreasing in survival. In other words, their risk of extinction is increasing. But from 1996 to 2008, which was until our study last year, the last time that red listing was done for all coral species, you can see corals are the most at risk of extinction. Next slide, please. Right, so what we did, we worked on a 5-year project with the IUCN. I was one of many people looking at all 892 species of corals on the planet. That's an awful lot of species, and it took, as I said, 5 years. The first paper we published was late in 2024, showing that half of Atlantic reef-building corals are at elevated risk of extinction due to climate change and other threats. So it was as you can see, lots of authors. This is big science throughout the Caribbean. We use satellite data, we use individual monitoring data. I did lots of work around Jamaica, but also did lots of coral species elsewhere in the Caribbean. And so what that means is that the relative extinction risk is increasing as time goes on due to the global warming of the oceans. Next slide, please. Just to give you some idea of the criteria, here we have on the left-hand side, for each species on the planet, you fit into either extinct in the wild or regionally extinct. The ones we need to be most concerned about are those critically endangered and vulnerable. And then we have near threatened, those of least concern, and at the bottom you can see where we just don't have enough data on the species. Within that, we have ways of measuring the risk extinction. It's a very rigorous process. We look at population reduction over 3 generations or 10 years, restricted ranges geographically, small population size and decline, very small population size, and the probability of extinction in the wild. We use lots of ways of getting the data together and finding out for each species. And as I said, 892, most of them in the Indo-Pacific. That's why we did the Caribbean first, because there are fewer species. But you can see, even in the Caribbean, very, very high probability of risk and extinction. Next slide, please. So just to give you some of the indicators that we look at for each species, we look at the depth range, the global abundance, restricted ranges, the vulnerability to disease. Disease, unfortunately, is particularly prevalent in the Caribbean at the moment, and it's also now gone into the Indo-Pacific. Some corals are better at recovery than others, and some corals are more vulnerable to ocean acidification than others. Next slide, please. So this slide shows the proportion of Atlantic reef-building corals in the red list by family. So we look at the families of corals and we look at the numbers of corals. So for example, at the very top we have the Acropora, two species of Acropora, and as you can see, they are critically endangered. And they are the fastest growing corals. They grow 10 times faster than the others, and it's a really serious problem. Then we have the plate-like corals, the Agaricidae, 9 species there, they're at risk of extinction, and so on, down to the least concern at the bottom. But you can see there how we've looked at a lot of criteria. Next slide, please. This is the distribution of threatened Atlantic reef-building corals in the ecoregions. At the top on the left-hand side, you've got a number of species, and you can see there in the Caribbean, on the left-hand slide, we've got a very large number of risk species to the east side, around the coast of Africa and the Mediterranean, where there are very, very few species. I mean, really very few species indeed. The map below that, B, shows the proportion of species threatened by marine ecoregions. So that gives you a proportion. More or less the same, pretty high proportion of species threatened, even higher, as you can see, around the east coast of Brazil there. Very serious from runoff, from pollution, and from disease. Next slide, please. What we tried to do, this is very difficult, look at the estimated change in coral cover by marine ecosystems and ecoregions. I mean, basically, you can see there the reefs around Brazil. The standard deviation is pretty wide, but over most of them, there is a reduction. That means the red and the blue go to the minus 100 rather than the plus 100. And that's really serious. It shows that although the uncertainty is pretty big there, the estimated changes, and you can see the median estimates in blue 1. T1, the blue one was to 78, T2, most recent one to 2019 is in red, and generally speaking, for each ecosystem, they're becoming more endangered. Next slide. So to summarize, 26 coral species out of the about 50 are now listed as critically endangered in the Atlantic Ocean, where almost half of all corals are now at elevated risk of extinction due to climate change and other impacts. I showed you a pillar coral, one of the slides recently. That has moved from vulnerable to critically endangered after its population has shrunk 80% since 1990. A very urgent threat is scaly coral tissue loss disease, infecting a large amount of reef per day. And you have there a summary: increased sea surface temperatures, excess antibiotics, fertilizers, and pollutants running into the sea have made corals more susceptible to disease. Certainly in the Caribbean, overfishing has depleted grazing fish, allowing algae, macroalgae, who are useless for ecosystems, to dominate. That's very true around the reefs of Jamaica, and putting further pressure on coral reefs. This is what I cannot emphasize enough. We need urgent action to address climate change impacts on both biodiversity and ocean ecosystems. So what can we do? Next slide, please. Next slide, thank you. So we know that things are very critical. We know that the last COP meeting didn't agree to reduce fossil fuel burning per se. So we have to look at research on really methods that we wouldn't use unless we were in such a critical condition. Basically, there are two of them. There's solar radiation management on the top, where you're trying to reduce the amount of sunlight coming down onto the planet. And secondly, carbon dioxide removal, where you're taking carbon dioxide, capturing it in the atmosphere, and burying it, for example, under the ocean. And there's quite a lot of research going on in that sphere. Next slide, please. We've done some work, this is with colleagues at Shandong University in China. We published that a few years ago, 2018, showing that if we inject aerosols into the stratosphere, then particularly sulfur dioxide particles, then you get a significant reduction in the ocean warming. And you can see here on the left-hand side, there's the Caribbean. You get a significant reduction in the warming if you do geoengineering, as opposed to, on the right-hand side, the representative concentration pathway, 4.5, which is a sort of a median reduction in carbon emissions. So in the Caribbean, this process would be very effective. It would also, in fact, reduce the amount of high energy hurricanes, because of course the water wouldn't be warming so much, and so you would not get that energy going up into the hurricanes. So let me now go on, thank you very much, to a paper we actually published last week in Nature Communications with a myriad of people. A lot of co-authors, there I am there. You can just see my name highlighted in blue. This was put out by the Chief Executive Officer of the International Engineering and Technology Institute, of which I'm president, and he very kindly said, congratulations. This is a major study, global study, of the first severe widespread bleaching event, which happened from 2014 to 2017. So it was a major event where the waters warmed not just in the Caribbean, but globally. Next slide. And you can see that here. There's the picture. You can see Australia, sort of halfway, a bit, not quite halfway, to the middle and the left. You can see America, North America and South America there, and between Australia and North America, you've got most of the Pacific there. Then you move into the Indian Ocean, the Western Indian Ocean, Eastern Indian Ocean, and on the far right, you've got the Atlantic, of course. And this is looking at heat stress from satellites. And the data that we use to get all this came partly from satellites and partly from individuals doing lots of measurements under the ocean. That's why it's such a myriad of people that were involved all over the world. And you can see there, the darker the colour, the more serious are what we call degree heating weeks. Now, degree heating weeks is a measure of warming. 1 degree heating week is a warming of 1 degree for 1 week. Or 5 heating weeks is a 1 degree for 5 weeks, or rise in 5 degrees for 1 week or something in between. So it's a semi-quantitative measure of looking at ocean warming, particularly near the surface, of course. And corals, because they need sunlight, generally live, certainly reef-building corals, in the first 30 or 40 meters. Though there are others lower down. So you can see there, we get serious alert level 3s right throughout the Indo-Pacific, between the west coast of the Americas and China and Australia, and then you can see Indonesia. The Coral Triangle, of course, is from the Philippines through Indonesia right through to Papua New Guinea. So very serious, and of course in the Indian Ocean there. So a lot of stress. Next slide, please. And to summarize, we analysed 15,066 individual reef surveys during this time, and 80% of them have experienced significant coral bleaching and 35% coral mortality. So once again, we have this really anthropogenic climate change causing absolute destruction to these extremely important ecosystems. And I give there a couple of recent papers. There's one that came out last week in Nature Comms, and then one that I also looked at, deep sea protection, in a book called The Fragility of the Ocean. Next slide, please. In our Nature Comms paper, we looked at the presence of heat stress throughout 3 time periods. The top one, A, is the one I've just shown you. B and C. B is from 2009 to 2011, and C is 1997 to 1999. And the insert of the left-hand side of the graphs show the relative proportion of degree heating weeks. And you can see by going from the 90s up to 2014 to 17, you've got a significant increase in the amount of degree heating weeks. And that's very, very serious. They're getting much more lengthy in the hotspots. And that's very, very serious for the future. So the satellite data comes really from NOAA, that's the National Oceanographic and Atmospheric Administration in the United States, in Washington, and they've had to increase their alert levels throughout that period of time. We are getting stronger and stronger global warming in the ocean. How anybody can say that it's not happening defies rational thought. Next slide. This is quite a complex slide. You've got the PowerPoint, so you can keep this, of course, and look at it in more detail. Basically, it's looking at projected coral reef bleaching and mortality impacts during this bleaching event. Basically, the darker, the worse it is. We've looked at both the northern hemisphere and the southern hemisphere. As you would expect, most heat stress in the Pacific, causing a lot of damage and a lot of projected extinction risk for these corals. And you can look there at predicted mortality, and predicted mortality has increased significantly during that period from the 90s to when we stopped this study. Next slide, please. And you can see that on this slide, looking at bleaching and mortality. You've got degree heating weeks in blue, greater or equal to 4 degrees, and in red, you've got very serious ones, greater or equal to 8 degrees. Remember that's 1 degree for 8 weeks or 8 degrees for 1 week or something in between. And you can see there is a tremendous increase in the amount of degree heating weeks during that period, and that significantly affects mortality. Next slide, please. So we then wrote our report for the IUCN, the International Union of Conservation of Nature. This happened in December 2024. Over 40% of coral species face extinction, and that's 892 species. Climate change is the main threat, and the main solution is cutting greenhouse gas emissions, accompanied by actions to address other threats, for example, pollution, all those runoffs from agriculture, to strengthen the resilience of the species. Next slide, please. So what do we do next? Well, there has been work on the policy effects of red listing species. This is, here we have a couple of papers. You can introduce legal changes. You can get recovery plans. You can change waste disposal. You can improve air quality. Have a climate change contingency and adaptation plans. And I think the essential outcome of the last COP meeting, was it in Egypt, was basically that nations have to do it themselves. Then we don't have global governance. That seems to have failed completely now. Even though China is trying to do some things in Asia, quite effectively, but we don't have global governance, so it's up to each nation to really put as many policy changes as possible in place to try and help the situation. Next slide, please. So the climate crisis, I think, has brought humans to a crossroads of survival and development, and it's incumbent upon us, incumbent upon you and me and all of us to break the deadlock. Carbon equality is very important because it starts from the balance of rights and responsibilities and directly targets the crux of carbon emissions. We need consumption-driven international cooperation and participation. We need to refine international use, overcome technological bottlenecks, and innovate social compacts. We need to work together and persevere in the implementation of carbon equality strategy to solve this crisis, protect the ecological environment around the Earth, and nothing less than determine the future fate of mankind. So I and others have written a couple of papers on that that you can see there. Next slide, please. So let's help each other across our cultures and beliefs. That is absolutely vital. Research should know no boundaries, and it's incumbent upon us all to work together to try and get much more equality, not just equality, but equity throughout our globe. Next slide, please. So, as T.S. Eliot, the great poet, said in his poem, Little Gidding from the Four Quartets, time past and time future allow but little consciousness. That consciousness is now. We need to work together to try and solve these problems. Thank you very much for listening, and to all my friends who I've worked with. Some of them are shown there. I'd like to highlight the one on the, particularly on the top left-hand side there, Alashna with us, who is a collaborator in China. Thank you very much indeed for listening.
Thank you, Professor Krave. On behalf of the organizing committee, I extend our sincere gratitude to our distinguished keynote speaker for an illuminating and thoughtful, provoking lecture. Your insight into coral reef sustainability, large-scale bleaching events, and the accelerating impacts of marine heat waves have given us both scientific clarity and a sense of urgency. Now, the session is open for any question and answer. If anybody have any questions to ask, please. Sir, I have a question.
Yes.
Your research integrates satellite monitoring, large-scale field surveys, and predictive modeling. What gaps remaining in global reef monitoring that limit our understanding of extinction risk for the estimated 44% of the threatened coral species? What gaps?
I must. The sound wasn't quite good. Do I understand your question to be what steps we can take to try and preserve these species?
Now, what is the limit of our understanding of extinction risk for the estimated 44% threatened coral species? What gaps are there? Because you have taken so many measurements like field survey, predictive modeling, and satellite monitoring. What else are there which maybe you can consider to know more information or something like that?
Okay, well, yes, of course, there is modelling. There's an awful lot of modelling that goes on. But the thing about our study that was published last week, the global study, is that was actually real-time monitoring, which we then use to predict what's happening in the future. And I must say, it doesn't look very good. Unless we get reduction of carbon emissions, then I don't see that the situation will improve. It's actually going to get worse and worse. And some of my colleagues say that we've actually gone beyond the tipping point, certainly for coral reefs. Coral reefs are the ecosystems most at risk of any on the planet. So they're a canary in the cage, if you like. And, they've been going down since 1990. Well, in fact, further back than that. And the reason is overfishing, the first instance, and of course now we've got global warming. All the fish are being taken out. So we need a lot to preserve the fish. Particularly the high predators, the top predators. A lot of top predators have just gone because there's nothing that they can eat there. And we need to get a complete ecosystem. So what we're doing at the moment with the IUCN, what I'm involved in, is red listing of ecosystems. So what that means, we take a region like the Caribbean, we divide it up into sections. For example, there's the Greater Antilles, there's the Lesser Antilles, there's the Florida Keys, and so on. And we look at the extinction risk of the ecosystem, not just species, but the ecosystem. And we started that process. The first ecosystem we looked at, Western Indian Ocean, published about a year ago in Nature Sustainability, and I'm working on the Caribbean with colleagues there. Very excitingly, we just the publication, which I was involved in, the current situation in Europe. We got used that also to get our whole ecosystem red listing going. So we're working hard on that ecosystem by ecosystem throughout the globe. Now that will give a globe picture, not just individual species, but of ecosystems, ecoregions, which are at risk of extinction. The Coral Triangle, which has the greatest biodiversity globally, as I said, Philippines, Indonesia to Papua New Guinea, is sort of the map, really in a hotspot of global warming. And if that goes, then all those people there who rely on reefs for their livelihood are going to be under serious threat of extinction themselves.
Thank you very much. And one more question. Looking ahead 30 to 50 years, under best case and worst case climate scenarios, how sustainable do you realistically believe coral reef ecosystem will be in next
Sorry,
30
I
to
didn't
50
quite
years?
catch.
Looking ahead 30 to 50 years, under best case and worst case climate scenarios, how sustainable do you realistically believe coral reef
Right.
ecosystem will be?
Yep. Got it. How sustainable with the best case and the worst case? Well, the worst case is that we are going to lose all the critically endangered species and all the critically endangered ecosystems. I don't think it's going to be quite like that. I've done a lot of scuba diving, and one of the reefs that is a very interesting one, it's one of the northernmost reefs globally, is in Bermuda. Now, Bermuda is far north than the tropics. So how do you get reefs there? Well, the reason is that you've got the Gulf Stream. You've got the warm water from the Caribbean coming up, and that is, as you know, it's the circulation, the meridional circulation system, and it's that warm water that has allowed some reefs in the Caribbean, some species to actually grow. But you don't have any Arctic corals there. You only have massive corals. And when I was going to dive there, I thought I would see a very depauperate ecosystem. In fact, it wasn't like that at all. You had very few species, but they were all growing very well. So they were working within their own limitations. But it was very, very different. And I think that's what we're going to see. Certainly by 2050, and definitely by 21, we're going to lose a lot of species. The gyre pillar coral in the Atlantic, 80% lost since 1990. You know, certainly going to lose that. That's a very beautiful coral. I mean, I've dived many years in the coast of Jamaica, and some of those corals I think of friends, and to lose those is so tragic. When you think of the bigger picture of how important they are for livelihoods of people who live there, and not just live there, but live elsewhere, that use the fish, that have invertebrates we use for medicines, and so on. It's so tragic that we are giving on ourselves. We actually don't need more research. We know what's happening. We need policy changes. And we need exactly the opposite of what's happening in the United States government. We need to replace, as Antonio Guterres, the Secretary General of the United Nations, said, we are in a situation where the law of power has overtaken the power of law. We need to get back there into what I would call, not just use separation of powers, where you separate the legal system from the policy system. That is so important. It's government. We lack government. We lack good governance in many countries, and we don't have global governance. So, these are the problems that we have to address. They're big problems. I'm all with the science and part of the policy engagement and so on. But if the politicians sort of go for increasing mining, extraction industries, deep-sea mining, it's just going to get worse.
Thank you very much. If there are any questions, not, then thank you very much. Today's lecture reminds us that coral reefs are not only biodiversity hotspots, but also parameters of planetary health. When nearly half of coral species face extinction risk, the question of sustainability becomes not only ecological, but ethical and global. We sincerely thank our keynote speaker, Professor Grave, for sharing expertise and global insights with us. The seminar has strengthened our understanding that coral reef sustainability depends on immediate climate action, robust scientific monitoring, and international cooperation. Let us carry forward this discussion into research, policy, and practice. With that, I formally conclude this session. Thank you all, and thank you, Professor Krape, for very excellent and very
Thank you very much. It's been a great honor and a pleasure.
Thank you, all the respected speakers and chairperson, for conducting this session smoothly. And it is a proud privilege for us that we have reached to almost at the end. And as you know, the 2 days of international conference is truly given us insights in the various aspects of biological science. And truly we have learned that how that sustainable development matters, and this ecological perspective, genomic science, and everywhere this the conjuncture between the subject and the amalgamation of the outcome of the interdisciplinary approach of the subjects will definitely enrich us, and truly we are overwhelmed and we are thankful to all the organizing committee. And as you all know, that sir was talking about the cultural integrity, and I found a great quotation from the Bhagavad Gita. আত্মৈব পেন সর্বত্র সমং পশ্যতি জহ অর্জুন সুখং বা যদি বা দুঃখং স যোগী পরম মত। হে অর্জুন, যিনি সকল প্রাণীর সুখ ও দুঃখকে নিজের সুখ ও দুঃখ বলে অনুভব করেন, তিনিই সর্বশ্রেষ্ঠ যোগী, এই আমার অভিমত। সত্যি তাই, and we are truly overwhelmed and thankful to our honorable principal madam. At a very short period of time we have tried to organize this seminar, and we are really looking forward for upcoming events, and Asansol Girls College is truly going to mark a significant contribution in the in the space of higher education. And it's my proud privilege to welcome the organizing secretary, Dr. Anand Sharma sir, who himself a great acknowledged scientist and has an illustrated career in SRM University, and we are fortunate once again that he has joined Asansol Girls College 2 years back, and it's a great moment to welcome him once again to say the formal voice of a vote of thanks.
Thank you, Dr. Arnab Chatterjee. Good evening, one and all present here. So today it's my privilege to propose a vote of thanks on this occasion of our international conference on Nexgen's Bioresources Frontiers for Sustainable Development, Sustainable World, sorry. So at the outset, I express my deepest gratitude to our respected principal madam for her constant encouragement, visionary leaderships and support. With this, in these 2 days of national conference, we have speakers from Brazil, UK, New Zealand, China, and Finland, and also from different parts of India. And we are fortunate to have the speakers like Professor Bhaskar from China, Professor Fred Duby from Finland, Dr. Millie Taylor from New Zealand, Mr. Pendit Traffordar from New Zealand, Dr. Jackson D'Souza from Brazil, Professor Michael James Scraby from Oxford, and Dr. Swati Nyogi from IIT Kharagpur, Dr. Rajat Pal from Sister Nivedita University, Dr. Mausumi Poddar Sarkar from Presidency University, Dr. Suparna Mandal Biswas from ISI India, Dr. Subarna Chowdhury from NIT Durgapur. So we have a very eminent speakers who have given their valuable time in these 2 days of international conference. My heartfelt appreciation also goes to all the members of the organizing team and the various committee members, and especially student volunteers whose dedication, teamwork, and tireless effort for this conference. A big thanks to all the speakers, the media persons for taking their time from their busy schedules to grace this event. Finally, I extend my gratitude to everyone who contributed directly or indirectly to this event. Thank you very much.